So welcome everybody. I'm Sid Gospe. Welcome to Grand Rounds. I'm not, that's Sid over there. Welcome especially to the applicants to our program. We have several applicants here. I did see some people wearing conspicuous name tags. So welcome, hope you like our program. My name's Hannah Tully. I'm in the Division of Pediatric Neurology. Sid is my boss and my colleague and my friend, and I'm here to introduce him. So Sid is from San Francisco originally. And he went on to get his Bachelor of Science and his Master of Science at Stanford. He then fled for the wilds of Durham, North Carolina, where he went to Duke, where he got both his MD and his Ph.D. Interestingly, his son, Sid Gospe III, also got an MD and a Ph.D. from Duke as well. I didn't know Sid. I was at Duke as well. I didn't know Sid then, but I did know his picture. There was a picture on the halls of Duke, a little tiny picture of Sid, and I used to walk past it every day. And when I was applying for residencies, I looked at that picture a lot and tried to imagine what kind of a person Sid Gospe was. I was partly right. He looks very similar to the way he did now. Same kind of intelligent look, same wry smile, same beard. But what was different in that picture was that he showed us that he has a real flair for paisley shirts, brightly patterned paisley shirts, very 1970s. And it's a shame because I hadn't seen this side of Sid since. But it's there, lurking somewhere. Anyway, after getting his PhD, Sid and his sartorial flair went off to Baylor, Texas to do his pediatric neurology training. And he was there with some heavyweights, Bill Dobbins, who's here with us now in Seattle, and also Huda Zogby. We're all there at the same time. And there are pictures to prove it. After completing his training, he was briefly in Albany and then was at University of California at Davis, where he spent several years. And then 14 years ago, we were lucky enough to get him here in Seattle, where he became the Herman and Faye Starkovsky Endowed Chair in Child Neurology, as well as the head of our Division of Pediatric Neurology. He's had a really amazing career. I knew this anyway, but looking at his CV was quite humbling. So I know that he's a great clinician and he has a focus in neuromuscular medicine. He's also a researcher and he's a renowned expert on pyridoxine-dependent seizures and formed a registry for that. He's well known as a teacher, and perhaps most impressive to me, he manages to do all those things and act as a magnificent division head, wrangling the members of the Division of Pediatric Neurology very well without ever letting us feel that we're truly being wrangled. He just manages to get things done. So those accomplishments haven't gone unnoticed. He's received several teaching awards. He recently, in 2014, received the Parker J. Palmer Award from the ACGME, and he was also inducted into the Gold Humanism Honor Society. So far, however, what's gone unnoticed is his developing talent on the baritone ukulele. So you'll have to ask him about that. Maybe one of these years that will make it onto his CV as well.
Can't play it right now.
So it's my pleasure, and I'm very much looking forward to Sid's talk, nicely alliterative, of manganese in men, trace metal neurotoxicity, and the dissection of a new disease.
Thank you, Hannah. That was quite the introduction. I really appreciate it. So I'm glad to be giving grand rounds today, and my goal today is to really take you through an approximately 25-year journey that I've had with a particular patient and his family, and learning about his condition and discovering along the way, along with many colleagues, exactly what was going on. So I have no conflicts of interest or disclosures today. And I'd like to start off with a brief reflection, and this is really a quote from Charcot, who had many great quotes, but this one I think is particularly pertinent to the topic that I'm going to be presenting today. Symptoms then are in reality nothing but the cry from suffering organs. And this is a very famous painting of Charcot teaching in his clinic, and there are many luminaries in this audience besides Charcot, including his student Babinski, Tourette, Marie, and Bournville, among others, are pictured in this painting. And one reason I show this is that Dr. Ransom, in some of his history of neurology talks that he sometimes gives in the summer each year, speaking of Dr. Ransom, he just walked in.
waiting for my.
Great timing,
Thank
Bruce.
you.
You're welcome. Anyway, Dr. Ransom sometimes will talk about neurologists in academics trying to trace their neurological pedigree. And a few years ago, I was able to do that through a website called neurotree.org, and found out that I was able to truly link myself directly to Charcot. He's my great-great-great-grandfather in my neurological pedigree. So this is through Phil Dodge at Washington University, who many people in the audience may know. So this particular painting has some definite meaning to me. So my outline today is I'm going to begin with a case presentation of a young man that I saw many years ago, over 25, 26 years ago, who presented with a gait disturbance and was found to have hypermagnesemia. I'll then talk a little bit about the biological roles and homeostasis of manganese, and then talk about clinical forms of manganese toxicity and the pathophysiology of what we know about manganese toxicity. Then go back to the case presentation and talk a little bit more about the further evaluation of this particular patient. And then we'll go on and talk about the discovery of the genetic basis of this patient's underlying disorder, which developed about 5 or 6 years ago. And then lastly, go back to the patient and talk about some long-term follow-up. And lastly, I'd like to end with a brief post-scriptum, which I like to call perseverance in publication. And that last part of the talk is really directed toward the residents and our applicants and some of our junior faculty. So I first met this fellow when he was 14 years old. I was probably in my first year on the faculty at UC Davis at that time. And he was referred to me by the pediatric hematology service. We had a history of malaise and stiffness of gait that lasted for about 2 months. And the hematologist and I basically only uncovered that he had some hepatomegaly and some increased tendon reflexes. Everything else was pretty much unremarkable, except his labs. And he had a markedly elevated hematocrit of 67% with normal red cell indices, a normal erythropoietin, blood gas, hemoglobin, oxygen dissociation curve. He had no evidence of malignancy, hemoglobinopathy, or cardiopulmonary disease. And that's what we knew when we first saw him. And he went off back to his home in Northern California, and his polycythemia was treated very successfully with biweekly phlebectomies. But over the course of the next year, his neurologic problems became much more significant, and he had leg weakness that was progressive, gait stiffness, and came back to neurology for a reevaluation. And when I saw him at that time, he had a definite spastic paraparesis, normal upper extremity function, no sphincter involvement, and normal cranial nerve function. He had this very nonspecific mild pinprick and cold temperature sensory loss. And then no other motor features, no cerebellar or extrapyramidal dysfunction. And again, he had some mild hepatomegaly, but nothing else on his examination.
Didn't look at all Parkinsonism?
Not at all, no, just a spastic paraparesis. Dr. Swanson asked if he looked at all Parkinsonian, but he had no movement disorder other than the upper motor neuron signs. His past history was a little odd, and it was pretty hard to actually document because it was all verbal history, but he had 3 hospitalizations between 5 and 12 months of age for what was termed aseptic meningitis, and this was in another region of California. He had some mild language delay, but nothing too terrible. And the rest of his early childhood development was normal. As far as his family history was concerned, there was no recognized history of consanguinity. There was no neurological or hepatic or hematologic disease in family members. He lived with his mother and his half-sister. His labs were, other than the elevated hematocrit, were really unremarkable, and you can see here that a large laboratory investigation was performed with serum chemistries, liver panel, vitamin studies, metabolic studies, a lot of rheumatologic studies, serologies, etc., and a CSF study, and everything was quite normal. So we got an MRI scan, and his spinal cord MR was completely normal, but his MRI of his brain was quite striking. And as you can see here, there was a really marked signal change within the globus pallidi of high T1 signal, This was also noted in the brainstem and the dentate nuclei in the pituitary, and there was some mild brainstem atrophy. And these signal changes were consistent with the deposition of manganese. So I was able to start my collaboration with some colleagues at UC Davis, and one of the collaborations that I had was actually with the Department of Nutrition, which coincidentally is not in the medical school but was in the agriculture school at that particular university. And a lab person there did a fair number of studies on trace metal and heavy metal assays, and was able to develop a specific manganese assay, and we measured this fellow's manganese level, and it was over 23 times normal. As you can see here, 3.48 micromoles per liter when the normal range was 0.15. So this was a massively elevated manganese level. So let's talk a little bit about manganese. Manganese is an essential trace element. It's involved in lots of different enzyme families, as you can see here listed. It's very important for the metabolism of amino acids and lipids, proteins, carbohydrates, and protein glycosylation. And very importantly, it's a component of metalloenzymes such as manganese superoxide dismutase, which of course is important for detoxifying free radicals. As far as dietary sources and the recommended daily allowance, manganese can be found in a wide variety of foodstuffs. If we go back out to the snacks outside, there's plenty of manganese out there. I made sure to have one of the little shrimp rolls, for instance, which certainly has some manganese content. And we can see that the recommended daily allowance of manganese ranges, at least in adults, around 2 milligrams a day. Slightly higher for a lactating mother. There is no report in the medical literature of somebody who was dietarily manganese deficient. One can certainly make people insufficient or deficient experimentally, but there have been no reports of humans who have actually presented with manganese deficiency on a nutritional basis. So the homeostasis and absorption of manganese is really quite complex, and it's incredibly tightly regulated. And here, if we look at the periodic table, we can see that manganese and iron are right next to one another, and this is an important concept to remember. The gut really controls the absorption of manganese quite exquisitely, and only about 1 to 5% of ingested manganese is absorbed. The excess manganese that is absorbed is excreted through the portal circulation and into the bile, and so it's easily disposed of. And there's this very tight connection between iron and manganese, in such that if a patient is iron deficient, you're actually going to have an increase in manganese absorption because the transport mechanisms are tied to one another. So the body will be trying to bring in excess iron, and in turn it's also going to be bringing in excess manganese. But in turn, if somebody has excess iron, that's actually going to reduce blood manganese due to similar shared mechanisms. As far as environmental sources of manganese and how you might absorb manganese through the environment, there are lots of ways that somebody might be exposed to manganese through industry or the environment. It's a very fundamental component in battery production. It's a major part of the welding industry, and welders can easily be exposed to manganese fumes during their work. Same thing with ferroalloy smelting. Manganese is part of that process as well. And manganese mining is a very common way that workers can be exposed to the metal. As far as how it actually gets into our system, other than just through ingestion, as I had talked about a few seconds ago, there's some suggestions that manganese may actually be transported to some extent through the olfactory nerve or the trigeminal nerve. So through inhalation through the nose or the face, there may actually be some manganese absorption in that fashion. There can also be transport across the pulmonary epithelium, and then if there's any mucociliary clearance in the nose, in the lungs, and swallowing of that contents, again, manganese can come in through the gut. So many different potential portals of entry. Now, manganese toxicity has actually been known for a very long time, and this is a photograph of a manganese mine in Gambia. The first description was in 1837 by Cooper, who described a Parkinsonian syndrome, which included tremor, gait disturbance, and a whispering speech in 5 manganese miners. And then this syndrome was much more extensively described in 1955 by Rodier, who described the syndrome in about 151 Moroccan manganese miners. There are actually 3 stages of manganism, and the first stage is one primarily of cognitive changes, emotional changes and cognitive disturbance, with some impairment of fine motor coordination. And then the second stage is much more profound, where patients have psychotic behavior, they have slurred speech, clumsiness, and mask-like faces. And then finally, in the third stage of manganism, these individuals have very marked limb rigidity, dystonia, impairment of balance, and this very characteristic cock-walk gait, as it's described. And these folks generally need a cane to be able to ambulate. Here you can see a gentleman on the top view who has that gait pattern, and in the bottom picture is a photograph of several South African manganese miners who have all been impaired. Now, manganism, the movement disorder for manganism, has many similarities to Parkinsonism, as Dr. Swanson was asking about potentially in my patient, but there are some distinct differences. First of all, there's a less frequent resting tremor, there's more frequent dystonia, there's a high propensity for them to fall backwards, and very importantly, there's usually a failure to achieve a very sustained therapeutic response to L-DOPA. And it's thought that this might be due to the fact that manganese, as far as its toxicologic function is concerned, may cause an abnormality of dopamine release without actual significant reduction in dopamine content or dopamine terminals, and hence the L-DOPA would be ineffective. Now, there are other causes of manganism as well, besides being a miner or a welder or working in the alloy smelting industry. And one of them would be exposure to certain gasoline additives, such as MMT, which is methyl cyclopentadienyl manganese tricarbonyl, which can be seen right here. And there have been examples of manganism that have been reported in people who've been exposed to this chemical. Another way that manganism has been produced is through the abuse of a drug called ephedrone or methcathinone, and this is a synthetic stimulant that is made from either ephedrine or pseudoephedrine. And when cooking ephedrine or pseudoephedrine to make ephedrone, there is an oxidation step that is required. And at least in Eastern Europe, the oxidizer that has been used most commonly is a permanganate oxidizer. And so the street drug ephedrone has been contaminated by manganese, and the individuals who have been using this stimulant have been exposed to manganese and have developed a manganism-like syndrome. The ephedrone that is cooked in the Western Hemisphere there's a different oxidizer step, and so the risk of manganism is not there. TPN is another reason that manganism has occurred clinically, and when somebody is on TPN, any amount of manganese that might be in the TPN formula is not going to be discarded in the same way as manganese that would be absorbed enterally, and this is because the TPN is bypassing the enterohepatic circulation system, so any manganese that gets into the blood is going to stay there primarily, because there's going to be a reduction in biliary excretion. Also, it's important to remember that TPN as a side effect can cause some degree of cholestatic liver disease, and so that too can reduce the actual excretion of manganese and lead to increased manganese levels. So long-term TPN use can certainly lead to increased levels of manganese in the brain with changes on the MRI and some extrapyramidal effects. And so in most circumstances now, manganese is left out of TPN formulas, even though there are some residual amounts of manganese that are there just as a contaminant. So for people who are on long-term TPN, it's still important to be following them for the potential neurologic consequences. Lastly, from a pure neurologic standpoint, I'm sure people in the audience have seen patients with what's called acquired hepatocerebral degeneration. These are people with very advanced liver disease, and for the same reason, as I mentioned just previously, they have impaired manganese excretion because of lack of appropriate portal circulation and due to the fact that they have portosystemic shunts. It's felt that about 1% of patients with cirrhosis are going to have some form of movement disorder, and many patients with chronic end-stage liver disease who are awaiting a liver transplant are commonly affected by some degree of hepatocerebral degeneration and will have a more significant movement disorder. Pathologically, the patients who've been studied neuropathologically with this disorder will show Alzheimer type 2 astrocytosis in the globus pallidus, and then from a neurological standpoint, clinically, these patients have dystonia, chorioathetosis, dysarthria, ataxia, pyramidal signs, and in rare cases, they can have a myelopathy, which is termed hepatic myelopathy. And I'd like to focus on that particular aspect of the syndrome. Just keep that in mind.
Sid, can I ask you about the non-Wilsonian acquired hepatocerebral degeneration? Neuropathologically, it had been the hallmark of that condition from a cytologic standpoint was the Alzheimer type 2 astrocyte. Are you suggesting that that's a manganese consequence, independent of the hepatic failure?
So Dr. Ransom is asking if the Alzheimer type 2 astrocytosis is a pure manganese effect or if it's partly due to the hepatic failure. I don't think I can honestly answer that. I think it's probably multifactorial. But there are experimental instances of hypermagnesemia that have also shown that type of change. So let's talk a little bit about the actual cellular mechanism of manganese homeostasis, and this gets pretty darn complicated, and I'm not going to dwell on everything in these illustrations, but I do want to point out a few things. As far as what's going on in the GI tract, manganese is primarily absorbed through this transport mechanism that you can see with these little red barrels right here, and this is called DMT1, or the divalent metal transporter type 1. This is a co-porter, not Cole Porter, the playwright and musical theater author, but a co-porter, something that transports two different ions, so a hydrogen ion and a divalent cation would be transported at the same time. So this can transport manganese, it can transport iron, and this is the way that manganese is transported into the enterocyte.
But not copper, is that right?
Not copper, no, that's correct. So that's how manganese gets into the enterocyte. As far as the way it gets out of the enterocyte, it actually leaves through this little yellow transporter here, which is called the iron exporter, also known as FPN. Once manganese gets into the bloodstream, it can be found as either a divalent or trivalent ion, and the divalent ion is bound to beta globulin while the trivalent ion is bound to transferrin, and that goes through the circulation and gets elsewhere, and primarily gets to the central nervous system. So once manganese gets to the brain, all of these other transporters come into effect. And DMT1 is also active in the brain, so manganese can get into neurons or glial cells through DMT1. It also can get in through another set of transporters called ZIP8 and CYP14. And then there are a large number of calcium channels that also can transport manganese as well. And then a manganese citrate complex can also be responsible. And then lastly, the trivalent manganese that's bound to transferrin can be internalized and get into the cell as well through the endosome. So manganese can get in a number of ways. Now, how does manganese get out? Because that's really important. And again, just like in the enterocyte, manganese can be effluxed through the iron exporter, FPN, as I mentioned before. It can also be sequestered in the Golgi through a couple of mechanisms. One of them is through a solute carrier, SLC30A10, and we'll spend more time talking about that in a little while, as well as can be sequestered in the Golgi through other mechanisms, SPCA1 and 2. And then lastly, there can be transport into the lysosome through ATP13A2, which is also known as PARC9. Now, from a neurogenetic standpoint, this is rather interesting because diseases have been described in several of these mechanisms. There is a form of hemochromatosis that is due to mutations in FPN. There is a very rare form of juvenile Parkinsonism that is due to mutations in PARC9, and as we'll see in a few minutes, mutations in SLC30A10 are responsible for the disease in the patient that I'm presenting. So let's get back to our patient. After we discovered that he had hypermagnesemia, my goal was to try and figure out why was this happening. I had no clue. So I was able to enlist the interest and collaboration of some epidemiologists through the California Department of Public Health, who are based down in the Bay Area. And they were able to make a home visit to this patient's home that was several hundred miles north of UC Davis, where I was working. He lived in a rather rural, small Sacramento Valley town that really had a surrounding volcanic geography. This was the southernmost range of the Cascade Mountains, which we know so well here in Washington. Nearby the home, there were no identified industrial or dump sites. And so the epidemiologists went to visit him, and they checked tap water at his residence and his former residence, checked the tap water in some of the surrounding homes as well, and manganese concentrations were completely normal. They did a 3-day food recall for the patient, and there was no obvious increased ingestion of manganese as far as his diet was concerned. Then they took some blood specimens and brought them back down to Davis, specimens from his half-sister, his mother, and the neighbor. And lo and behold, his half-sister and the neighbor had normal manganese levels. His mother had a slightly elevated manganese level, about twice normal. We did some other imaging studies to see if there was any abnormal signal change in other organs, and an MRI was done of his chest, abdomen, and pelvis, and all we saw was some hepatosplenomegaly, which we expected to see given the size of his liver on palpation. But there was a slight diffuse increase in T1 signal in his liver that suggested that there might be some metal deposition. He eventually had a liver biopsy, and he had micronodular cirrhosis. Copper deposition was not noted by the pathologist, but no other specific stains or studies were done in that consequence. And then my colleague in nutrition was able to get some liver tissue and measured tissue liver content, and it was 3 times normal. So additional history was obtained, and there was really no history of jaundice or exposure to hepatitis or alcohol or hepatic toxic drug use. He had no Kaiser-Fleischer rings. He had a complete GI workup and to evaluate for any suggestion of portocable shunting, and there were no collaterals found, no varices, etc. Again, more and more serologies were run to see if he had any past exposure to hepatitis viruses, and those were negative. Only an EBV serology that was consistent with a past infection, which is pretty inconsequential. So our working diagnosis was that he had a cirrhosis of undetermined etiology, and that he had a secondary hypermagnesemia, polycythemia, and what we termed a hepatic myelopathy. As I mentioned, hepatic myelopathy is a known consequence of chronic liver disease. It had never been described in a pediatric patient as far as we knew, but that was our best way of putting this together some 23, 24 years ago.
Said that in the other cases of manganese toxicity, do they get polycythemia?
No, they don't. And certainly nothing to this extent. So in 2000, I moved up here, I kept in touch with this patient and his mother and his half-sister. In 2008, in 2000, we eventually reported this patient to the medical literature, described his syndrome in our evaluation. Lo and behold, in 2008, a group in Britain published a series of two...
Can I ask you one other question?
Certainly.
At this point? Was there any consideration of doing some kind of chelation therapy?
Yeah, Dr. Bird is asking about chelation therapy. I'll be getting to that in a few minutes, so thank you. So in 2008, a group in Britain described two siblings who were the offspring of first cousin parents. And these children presented fairly early in life with extrapyramidal dysfunction of dystonia primarily. They had overt liver disease, polycythemia, and also hypermagnesemia. And we can see the MRI scan here on one of those two kids with very clear deposition of manganese in the basal ganglia, in the cerebellum and brainstem. So this really led to very important genetic investigations by actually two different laboratories, the group in London in Peter Clayton's lab and his postdoc Karen Tuchel, and then another group in the Netherlands led by Quadri were both working on this problem. And simultaneously in 2012, reports were published in the American Journal of Human Genetics describing mutations in SLC3810, the Golgi sequestering enzyme that I mentioned a few minutes ago, as the cause of this disorder. Whole genome mapping was done. It identified this gene on chromosome 1q, and at least in Tuchel's group, they were able to describe homozygous mutations in this gene in 8 particular cases. This kindred, another kindred, and then 6 other families, including my patient, were part of that report. And then Quadri simultaneously reported similar mutations in 2 other families. So a total of 20 affected patients in 10 families were reported. Tuchel's work reported 7 point mutations, primarily in exon 1 of this 4-exon gene. She also reported 1 multi-exon deletion, which spanned both exon 1 and part of exon 2. Quadri, on the other hand, reported 2 mutations, one of which was in exon 1, and then also one way down at the end of exon 4, which is not shown in this particular figure. All of the mutations that were described in these 10 families were homozygous. So SLA3810 was originally felt to be a zinc transporter when it was first discovered many years ago. But expression studies that were done by both of these labs definitively showed that it does play a role in manganese transport. And again, you can see that manganese and zinc are relatively close to one another along the periodic table, so we're going to share some mechanisms. The SLC3810 protein is a 485 amino acid protein. It's a transmembrane manganese transporter expressed in liver, small intestine, and brain. And importantly, of all of the 10 mutations that have been described, they all occur in highly conserved areas of the protein. So as far as the expression studies that have been performed, just to prove that this is a manganese transporter, Tuchel and her colleagues in London were able to utilize a specific yeast strain called Delta PMR1. This particular yeast strain fails to grow in high manganese environments. And they were able to develop a vector that could incorporate SLC3810 into that yeast strain and recover its ability to grow in a high manganese environment. And then they incorporated a couple of the different mutations that had been discovered in these affected patients and showed that the SLC3810 no longer could rescue the growth of this mutant yeast in a high manganese environment. So here you can see a number of wells with yeast growing quite happily in a variety concentrations of yeast in a normal media. On this side are the yeast that are growing in the high manganese environment, and you can see that if the yeast is given a particular, this is the yeast strain without the mutation, so it can grow in a high manganese environment, and then down here we see various other parts of the experiment where the yeast either got the vector without the rescue gene, and so the growth is much reduced or absent. And then when SLC3810 is added to the yeast, the growth is recurs, but when one of the various mutations is added in, the rescue is not as effective. Studies that were done by Quadri's group also demonstrate the effect of manganese on the expression of this protein. And here we can see that in a high manganese environment, SLC3810 is expressed, cDNA is expressed quite robustly, whereas if the cells are grown in a high zinc environment, the expression is not induced. And then looking at this western here, we can see that the SLC3810 protein is also detectable when it is grown in a high manganese environment. So what's the pathophysiology of this disease? Well, seems like the absence of SLC3810 leads to a decrease in manganese sequestration in the Golgi and cytologic manganese toxicity. It's certainly possible that manganese may promote erythropoietin synthesis and lead to polycythemia and reduced iron stores. The homeostatic regulation of manganese and iron, as I mentioned previously, are very tightly linked. And so, when manganese goes up, there's going to be a reduction in iron absorption and an increase in iron excretion, which can also lead to that imbalance. And as I mentioned, manganese and iron are very structurally similar, and they're competing for these binding and exporter proteins. This leads to a justification for actually treating patients with this disease with extra iron as a way of trying to reduce manganese absorption. And as Dr. Berg was asking a few minutes ago, what about chelation? So we'll get into that in just a minute. So again, I mentioned there's been now 20 patients from 10 families have been described with this disease. And we now know there's some degree of phenotypic variability. There's a childhood onset form of this disease where movement disorder begins between 2 and 15 years. In all the patients except for the one that I described, they present with a four-limb dystonia. And they usually have this very characteristic high stepping gait or a cockwalk-like gait. They have dysarthria, fine tremor, and a bradykinesia. My patient never had those symptoms, but only had spastic diplegia. An adult onset form has been described in a couple of kindreds, and these are individuals who present in adulthood with Parkinsonism, shuffling gait, rigidity, bradykinesia, etc., and unresponsive to L-DOPA. The liver disease is surprisingly variable, and some of the patients who presented early on in childhood had a relentlessly progressive cirrhosis, which led to death in adolescence or early adulthood. There are some patients who just have hepatomegaly and abnormal liver studies, and there's some who have no hepatic involvement whatsoever. And there's really no good explanation for this. As far as genotype-phenotype correlation, it's really hard to form any conclusions at this point, given the fact that there's so few patients who've been described. The one pathologic variant that Quadri described, the abnormality in the sort of end part of exon 4, leads to a frame shift abnormality due to a premature stop codon. And so in that protein that is synthesized, there is abnormal protein that lacks the last 49 amino acids. And those patients actually have this adult onset Parkinsonian disorder. So it's certainly possible that that truncated enzyme has very reasonable residual function, which allows the patients to live for many decades before they start having neurologic dysfunction. So what about the treatment of this disease? Well, The group in England, when they described these two kindreds, decided that it would be reasonable to try chelation to see if that would be a way of ameliorating this disorder. So they came up with a protocol of providing these patients with an EDTA infusions. They would treat them for 5 days each month. And when they did that, they noticed that blood manganese levels did go down. There was some improvement in their dystonia, and their brain MRI improved slightly. And if we look at these top two MRI scans here, we can see this one patient who was 12 years old when her treatment began. You can see the very clear manganese deposition in the basal ganglia and the cerebellum. And then after some initial treatment with EDTA, there is a subtle decrease in the MRI appearance of the manganese deposition.
Can the
This-
radiologist be specific that that's manganese, looking at those?
So Dr. Swanson is asking if the radiologist can tell that this is specific for manganese. I think it is a presumption that it's manganese, just known from past patients who clearly had manganese exposure from mining and from industry. I'm not sure if other trace metals could potentially cause a signal change like that. I don't know of any others that have been reported. So what these authors in Britain decided to do was actually to enhance the chelation therapy. They increased the chelation up to 8 days a month, and they added some iron as a way of slowing manganese absorption in the patient. And this was fairly dramatic. The blood manganese levels went down significantly, the brain MRI improved, and the patient did better, and liver histology also improved. This one particular patient who was treated in this fashion unfortunately left England and went back to her home country, where there were a lack of supplies for this aggressive form of chelation, and so her infusions necessarily had to be reduced in frequency, and her status deteriorated. So here we can see in panel C what her MRI scan looked like after the enhanced chelation therapy, but then once the chelation therapy went back down to a less frequent infusions, you can see that the amount of deposition increased again. She then was able to reinstate the frequent infusions, and over a number of years had marked improvement of her clinical status and really a quite nice normalization of her MRI features. So fortunately, there's some good examples of this patient's syndrome and her improvement that are available through the journal Movement Disorders. And the videos that I'm going to show you for the next few minutes come from that publication. So this is an example of how this child looked at presentation when she was 12 years old, before she had any treatment. And you can see that she's got some dystonic posturing of her fingers, quite slow movements as she's trying to do finger opposition. Her gait is very abnormal, with sort of a high steppage gait. I'm not sure this is necessarily a cock walk, but that's what her gait looked like at that time. Again, she was treated initially with 5 days per month of chelation, and then the chelation was increased to 8 days a month, and also with iron. And this is after 2 years of therapy. And you can see her movements are a little better. She still has some posturing of her fingers. It's kind of hard to see her attempts to make a spiral here. And her walking, I think, is a little faster. And then lastly, as I mentioned, she returned to her home country. Infusions were able to be continued after a while at a very robust pace. And now 9 years after treatment began, we can see how she looks. Marked improvement in her posturing. Good finger to nose. Rapid alternating movements, finger oppositions, not too bad. and her walking is pretty darn good. So the question is chelation actually helping her? Was that the question? I think so. Because that's the only treatment she's getting.
I thought she wasn't getting it for 9 years.
No, she was getting it for 9 years. She moved back to her home country. The chelation had to be ratcheted down for a while, but then it went back up to the more aggressive treatment. So, absolutely.
Are the ethnic origins quite variable?
Yes, they are. The 10 families described, they come from the Middle East, Sacramento Valley, the Philippines, Brazil, maybe one other area.
And consanguinity in some of them?
They're all homozygous mutations and definite consanguinity in several of them. So, what happened to my patient? Well, as I mentioned, I kept in touch with him and his mom and his stepsister over the years after I moved up here to Seattle. My colleagues in adult neurology were following him. He was primarily followed by hepatology and the hematology service. He developed pretty significant painful extensor spasms and got treated with intrathecal baclofen, which was really very, very successful for him. His imaging studies were quite stable. He had no change in his mental status. He had no development of extrapyramidal or other motor dysfunction. His hypermagnesemia was persistent. It was massively elevated. We checked his erythropoietin levels a number of times to see if there was any correlation between his manganese levels and the EPO levels. They were all over the map. Sometimes his EPO levels were just absolutely sky high. Other times they were normal. But his polycythemia persisted until he was 28. And then his polycythemia just went away. No good answer, but he no longer required bi-weekly phlebectomies. His liver remained irregular and firm. He had absolutely no stigmata of liver disease. His liver function studies were always normal. Attempts were made to get him into a chelation program. I had him, again, I was working long distance with this, but I was able to get him evaluated to start a chelation program, but it was, transport of him to get that started was very challenging. His mother eventually passed away, and he had to go and reside in an adult care facility. He was still quite functional. He had a volunteer job, was able to take the bus around town, and was very independent, but getting down for chelation was just not something that could be facilitated. So he unfortunately did not have a happy ending. At 38 years of age, he developed a very serious pneumonia and respiratory failure. He elected to withdraw support, and he expired. He had previously expressed an interest in donating his remains to research, and I had been in touch with his sister, who is half-sister, and we were able to arrange for an autopsy. And what I'd like to do now is present those autopsy findings. So from a liver perspective, he had very obvious hepatomegaly with micronodular cirrhosis. He did have portal hypertension and with congestive splenomegaly and very obvious esophageal varices, but they were never symptomatic. On his liver pathology, there were rhodamine positive changes in hepatocytes. The rhodamine is this sort of golden deposits that we can see here on this particular section. On the H&E, there were actually some brownish granules that were also in the same place as the rhodamine positive granules. And rhodamine is a stain that will stain both copper and manganese. We did Western blot analysis of liver protein looking for SLC3810, and as you can see here, it was absent. As far as the brain is concerned, there was very marked abnormalities in the basal ganglia with yellow-gray modeling of the globus pallidus and marked neuronal loss. There was loss of pigmented neurons in the substantia nigra, the dorsal motor nucleus of the vagus and locus ceruleus. There was a very marked activation of microglia and reactive astrocytosis, myelin loss and spongiosis in the globus pallidus. There again were these brown pigmentary changes seen in the neuropil and the few residual neurons of the globus pallidus, and this was also rhotamine positive, consistent with manganese. And this was also found not only in neurons but astrocytes and macrophages. We did some staining for SLC3810 using an immunoreactive stain, and in the residual globus pallidus neurons, we did not see any immunoreactivity, as you can see here on the right, and this would be a normal control section, proving that the protein is not present. Besides the figures that I've shown you, he had a reactive gliosis throughout his white matter. He had diffuse axonal loss in the cortical spinal tracts throughout the entire system, from the rostral cortical spinal tracts all the way down the spinal cord. There was no substantial reduction in his neurons, though, in the pars compacta or reticulata, the substantia nigra, despite the loss of pigment. Again, my colleague in nutrition was able to do some manganese studies for us, and was able to demonstrate a 16-fold increase in basal ganglia manganese and a 9-fold increase in liver manganese compared to controls. So we still have some unanswered questions about the pathophysiology of this disease, even though we now know what the genetic basis is. And one question is, why is hepatic involvement so variable? We know that in animal models, manganese promotes cholestasis and hepatocellular necrosis, so why do some patients go along happily without any obvious liver disease? The protein SLC3810 is expressed in the liver, so shouldn't it be clinically deficient in all of these patients? But we just don't know. Why did his polycythemia resolve? That was pretty striking, given how huge a problem that was when he first showed up at 14 years of age and for the next 14 years. And why, if erythropoietin is somehow linked to the polycythemia, why wasn't there some suggestion of correlation between EPO levels and manganese concentrations and what have you? And then, importantly, from a neurologic perspective, why are these patients with the extrapyramidal syndrome so unresponsive to L-DOPA? The unanswered questions in this disease are, first of all, are the dopaminergic terminals actually reduced in number? Are the dopamine levels normal, or are they reduced? Or is there actually an abnormality in dopamine release? And that's actually a mechanism that's been hypothesized in experimental models of manganism. So as I mentioned at the onset, I'd like to end with a brief post-scriptum about my 25-year journey with this young man. And again, I'm sort of now focusing on our residents and our applicants and some of our junior faculty. And what I'd like to, I'm not a motivational speaker, so I'm not going to try and get up on that podium, but I think it's important to always keep a very inquisitive mind about your patients and try to keep track of them. I don't mean keep track of your patients like Charcot did, and for those of you who don't know the history of Charcot, he actually hired some of his patients to work in his home and sort of kept track of them along the course of their disease because he could watch them every day. I'm not suggesting that we go that far, but with the internet, with email, with video, it's very easy to keep track of patients now as opposed to in the past. And when you've got an interesting patient that doesn't make a whole lot of sense to you, keeping track of them may help you out. You've got to search for collaborators because they're out there, and my ability to work on this for the better part of 3 decades was really to find epidemiologists, nutritional biochemists, geneticists, and neuropathologists. It was not that hard to get people interested in this very perplexing and interesting problem. And as far as publishing is concerned, all I can say is don't give up. The first time I tried to report this case, I should say the first 2 times I tried to report this case, the paper was rejected by 2 prominent pediatric journals. And interestingly, the very first title I gave this paper suggested that this was an inborn error of metabolism. And you can see that we called this paper Progressive Spastic Paraparesis Associated with Polycythemia and Cryptogenic Cirrhosis, a Possible Inborn Error of Manganese Metabolism. This was summarily rejected by the reviewers as part of their critique of the paper. And so we had to shelve that idea, and eventually the paper was accepted in the Archives of Diseases of Childhood, which is a British pediatrics journal, and that's where it was in 2000. But suffice it to say, I've been able to actually get 4 publications over the course of about 12 years in working on this problem and working with some really wonderful colleagues, geneticists in Britain, neuropathologists down at UC Davis, and then a gene review that Tom Bird asked us to write. So it pays to be to persevere and to work on something interesting. So I'd like to acknowledge my patient and his family. I'm still in touch with his half-sister. My colleagues at UC Davis, Mike Clegg, Marna Lischbaumer, and Li Wei Jin. My colleagues at the Institute of Child Health in London. And then Tom, who was kindly was a mentor and a cheerleader for me on and off over the last 10 years. I appreciated your enthusiasm as I worked through this, so thank you very much. Dr. Bird.
So the mother had an elevated manganese level. What do you make of that? And have carriers in other families had elevated manganese levels?
Right. So Dr. Bird is asking. My patient's mother was. We saw that her manganese level was twice normal, and his half sister was probably normal. And Tom's hypothesis is correct. There are a couple of other obligate heterozygotes who do have slightly elevated levels of manganese, not pathologically, but above normal. So I think that is a marker of the heterozygote state. Phil.
Would oral chelating agents be at all useful?
Boy, I'm not an expert in that. Phil is asking whether oral chelating agents would be useful. The only protocols that have been published have been this intravenous protocol. I just don't know. Bruce.
Sid, that was absolutely a terrific talk, and my hat's off to somebody who can be that persistent. I'll remind myself of that the next time you ask for something, and then ask again. I'll realize it'll go on until I finally say yes. The adult onset patients, probably not a lot of them, do they wind up having the same characteristic MRI pattern of change that you see in the younger patients, which you showed us?
I believe they have the same pattern of basal ganglia deposition in the globus pallidi, yes. I would have to look at those reports, but my recollection is that they do.
And if I could add one
And I'm sorry, Dr. Ransom was asking whether the few adult onset patients described by Quadri et al., whether their MRI changes were similar to the younger onset patients, and I think that is true.
So this struck a chord, and it comes at a timely moment. I'm in the process of treating a welder who has, from my eye, absolutely typical Parkinson's disease, but he has been very unresponsive, maybe I can say totally unresponsive to levodopa, and it's been a shock to me. So of course I'm aware of the fact that he's a welder, but he works for a big corporation locally that makes airplanes, and has a very high sensitivity to the conditions under which their mechanics and welders, etc., operate. So I never really thought in the modern era that one could acquire a welding-related manganese pathology. Am I wrong about that?
Well, Dr. Ransom is mentioning that he has a patient with a DOPA non-responsive form of Parkinsonism who happens to be a welder, and is questioning whether there could be a link there. And has that patient had an MRI?
I think so, without really anything much to see.
So I think it's certainly something to look into. With appropriate industrial hygiene, theoretically he's got good prevention of inhaling manganese fumes, and the risk would be relatively low. I think in people who do welding in less protected environments, independent welders who go out and do particular jobs, outside of a protected factory, I think the risk would certainly be higher, and one would hope the environmental protection of the employer that you are telling us about is pretty rigorous.
De-identified.
De-identified aerospace
I have a
company.
patient that was a steel cutter at a company that there was clearly manganese in the steel. He developed, again, quite severe Parkinson's disease, who, however, does respond. I mean, he stopped working, but he responds very well to carbidopa. But there's no manganese in his system at the present time that he needed.
So Dr. Swanson mentioned that he cares for a patient who works in an iron industry, who has Parkinsonism that is dopamine responsive and does not have evidence of high manganese. So I think it's just unrelated.
Well, it's thought to be related, at least by the industry, who's paid by L&I.
I see. So he's
His care, and
okay.
the people in environmental medicine think it was due to manganese exposure.
Dr. Bird.
So, two things. One, the manganese miners that are intoxicated, they sometimes later on, you said, got psychosis and behavior cognitive problems. Has that happened in any of these families?
Not to my knowledge, but there's certainly, some of the experimental studies of manganism in animals certainly show that there can be changes not only in the basal ganglia but also in prefrontal cortex, and there's a lot more going on than just in the extrapyramidal system.
And the other suggestion would be in some of the living people with this disease to do a DAT scan as a way of looking at the dopamine.
Exactly. So that I'm actually in touch with somebody at Columbia who, would like to get a patient with this disease over there for a PET scan, and he asked me if I knew of any other Americans with this disease, and I know of one other who happens to live on Guam and is a child of a consanguineous parents who are from some other very small Pacific island, and he's cared for by a child neurologist in Hawaii, and I was able to get his DNA and send it to Britain, and he's now another patient with known homozygous mutations. So he's the only other American patient I know of who's been diagnosed with this disease.
Sid, I think you answered all of the interrogators, so thank you so much for a great presentation.